A very good morning, class 12 students. Now I have switched on to another very important chapter that is about principles of inheritance and variation. This is one of very important chapter for every kind of either you talk about your examination, any kind of examination, board examination or NEET examination. This chapter is really very important for you to understand. So this is our chapter number 5, that is about heredity and variation. As the name is suggesting, that we are going to talk about genes, we are going to talk about characters, we are basically going to talk about first Mendel's work. So principal basis of inheritance. First, as you can see, that base of inheritance. What is the base of inheritance, due to which characters they are transferred from one generation to another? So first of us, for us to know the definition, in very important for us to know, that is about heredity and inheritance. That what is the meaning of this word? It is a process by which characters, or sometimes there is another word we write, that is the characters or traits, they are transferred from one generation to the other. What is the one generation? Here he is talking about parents. And where these characters they are transferring to the next generation. What is the next generation? That is the offsprings. So when we say that characters or traits, they are transferred from one generation, from parents to the offspring, which is next generation, the process through which they transfer, that is called heredity. Now, as we are saying, these characters, they are transferred. In sexual reproduction, we know that how many parents they are involved. Two parents are involved in sexual reproduction. So when their characters, they are transferring in offspring, they give rise to changes. So differences or different changes in the traits, or you can say characters like, if we talk about color of eye, or we talk about shape of chin, and shape of chin, then your color of hair. So different character, even through the genotypic differences also. So those changes in the characters of individuals, of a progeny, from each other means from their brothers and sisters also, and from their parents, they are called variation. मतलब आप में जो changes आ रहे हैं, आप में और आपके brother sister भी, they are not identical. You are not identical to your siblings, as well as you are not identical to your parents. So these changes which are observed, these are called variation. So two definitions need to be very clear here. One is the heredity and other is the variation. Whenever we talk about genetics, one thing we need to talk about is the scientist, that is Mendel. Without Mendel's work, we cannot study genetics in detail. That is why this person, this scientist, he is Mendel. Sir Mendel is known as father of genetics. He was born on 22nd of July 1822. For 6 to 9 years, he worked continuously on pea plant. Scientific name of pea is Pisum sativum. He died in 1884. But the question is, till he died, his work was not recognized. Still, even in 1900, his work was not recognized. 16 years after his death, three scientists they rediscover Mendel's work. So in 1990, three scientists, like scientists, Hugo de Vries, Correns, Shermark, they rediscover his work, and they gave recognition to his work. So these three scientists, they actually studied his work and they gave recognition. But till Mendel was alive, his work was not published and recognized. Why Mendel's work was not recognized that time? Work was not published properly, at that time actually Darwin's work was very famous that time, so biologists they did not accept Mendel's work due to some reasons. Factors, the word factors which he gave, it was not clear and understood by others, that how they passed on to from one generation to another. This was not able to understood by others, that how this is passing on from one generation to other. He called these factors, these are the discrete units, but these days we call them as the genes. So there were these reasons that why his work was not recognized at that time. Reasons, then Mendel, because he continuously worked on one plant only, that is the pea plant, he worked on Pisum sativum. What was the reason? Why did he choose this pea plant for his sample study, for his as a sample for his study? First thing about pea plant, very important, is its life cycle is very short. Just within 1 week you get another generation. Within 2 to 3 weeks you get another generation. Means 3 to 4 weeks that it takes for the another, for the new generation. So many generations they can be studied in very short duration. That was very plus positive point about pea, that life cycle is very short. Pea plant has bisexual flowers. Bisexual flowers कौन से होते हैं, which has both male and female parts. So it was one of the other thing to study very in detail. Mostly flowers they were self pollinating. So when flowers they are self pollinating, you know that you will get pure lines. Pure lines होती हैं कि means same characters you are going to get for several generation. Suppose if the color of the plant is purple, and I am selfing it with the purple plant, will only get purple. There will not be other thing. So same generation will be the, so several generation pure lines we will pure lines I will explain also to you, that what is pure line. That pure line is the same type of characters, like this is going you to, because there is no heterozygous, so there is same genes, as you can see capital T, so you are going to get it same for several generations. It is easy. Now self pollinating to ye hai dominantly, but you can use it for the cross pollination also. What you have to do, emasculation we discussed in the second chapter, that you can remove the pollen from and you can bring the pollen from another plant, and you can introduce the new pollens. You can introduce the new pollens from another plant by removing the anthers of the same plant. So this was called the emasculation. So cross pollination was also you can perform here. It was easy to perform. Pea plant was easy to raise, easy to maintain, easy to handle. That is why he chose this plant for his sample. Pea plant shows contrasting characters. Contrasting characters कौन से होते हैं, जैसे कि tall and dwarf, round and wrinkled. This was one of very important point about pea, that it shows contrasting characters. So that is why, and out of these contrasting characters, Mendel selected 7 contrasting character for his study. So these were the reason that he chose pea plant for his study. The seeds they do not show dormancy, means जैसे the moment seeds formed they will germinate. There was no resting period. There was no resting period. As soon as you will put them in soil they will germinate. Pea plants produce large number of seeds. What is the purpose of large number of seeds? That large number of seeds per generation you can get more and more sample size. More sample size means you're easy to collect statistical data, easy to basically collect your data for the study. So that was the reason that Mendel chose pea plant for this study. Now what were those 7 contrasting characters which he selected? First, if we talk about height, it can be tall or it can be dwarf or short. Dominant, I have written here, that is this is trait or characters he chose, and which was dominant and which was recessive. So he chose these contrasting characters are like, one is tall, another will be short. As you can see, one was tall height and another was short at height. Flower color, if we talk about, few they were purple and few were the white color. Pod color was, this was the pod color, pea pod, that is the green, and in few it is yellow. Seed shape is also, but yellow was recessive because it was observed in very few cases. Seed shape, mostly they were round, few were wrinkled also. So if we talk about seed color, the dominant was the yellow and green was recessive one. Flower position, it is axillary. As you can see, this is at the sides also, that on the branches also, that is the axillary position, means on the side position also. But terminal we use for the end. So flower position can be axillary also. Axillary was the dominant, that was the most commonly observed, and terminal was the recessive position. So pod shape is inflated होता है कि इट्स जैसे बोलते हैं ना हम, फूला हुआ बेसिकली, इट्स लाइक प्रॉपर इन्फ्लेटेड. नॉर्मली जो आप पी देखते हो, दैट इज इन्फ्लेटेड वन. एंड कॉन्स्ट्रिक्टेड मीन्स पिचका हुआ. एज यू कैन सी देयर आर मैनी रिंकल्ड ऑन द पॉड शेप. सो दिस इज कॉन्स्ट्रिक्टेड वन, एंड दिस इज द स्ट्रेट वन, एंड इट्स अ लिटिल like a proper with the seeds, and it is constricted. So inflated was the dominant. So these were the 7 contrasting character which is discussed by Mendel. Now there are few important terms in genetics which when we talk about the going to talk about the main chapter, these terms should be clear to you. Each and every term play a very important role. First, let's talk about gene. What is gene? If we talk about basis of inheritance or functional unit of inheritance, then that is gene. Without genes, if there will be no genes, then there will be no transfer of the characters. Term gene, that was given the name gene or term gene because Mendel called them as a factors. The term gene was given by Johnson. It was called factors by Mendel. So gene is clear. Next is the alleles. Now this gene, as we know that this gene has 2 forms. We always write gene as the double form, means that is, if this is the tall character, then this is one form, this is the another form. Basically it's copy. So alleles, these are the alleles are the 2 forms of a gene. If suppose for tall height my gene is T, how I write them in the form of allele? It is capital T. Or if it is heterozygous, then it will be like this. So there are 2 forms of a gene. If it is TT, then dominant is represented by capital and recessive is represented by short, that is T. So this is represented by the short or the small t. So these are 2 forms of a gene. You need to understand these alleles very clearly, that what are these alleles? They are 2 forms of gene. One gene, this is gene, it has 2 forms. These 2 forms, they are called alleles. Now homozygous, either both the genes they are dominant or recessive. If they are dominant, this is homozygous. If they are recessive, still this is homozygous, because those both alleles or both the alleles, they are copies are same copies genotypically, that is the homozygous condition. Heterozygous matlab both the alleles they are different, TT, one is dominant, one is recessive. This kind of condition is known as heterozygous. This kind of condition is known as homozygous. Because जब हम मेंडल का क्रॉसेस स्टडी करेंगे, देन वी ऑलवेज, जब हम जीनोटिपिक और रेशो लिखते हैं, वी ऑलवेज से दैट दिस इज homozygous, this is heterozygous. So for you, these terms, they need to be very very important to be clear, that this is heterozygous condition and this kind of condition is homozygous. Now next is the phenotype and genotype. As you can see, phenotypic characters. Phenotypic characters कौन से होते हैं? Which we identify as the physical appearance or just from the outer look. Genotypic, which are your genetic makeup, in which we talk about particular genes. Suppose, like example, now if I just look at it from outside, for me, this plant is tall, because I'm not looking at the genes or I'm not looking at the genetic makeup. But phenotypic, if I will say that this plant is tall because T is dominant over small t. But genotype, if I have to write this, I will genotypically I will write this as heterozygous tall. As you can see, this is heterozygous tall genotypically. This is height of the plant is tall phenotypically, but genotypically this is homozygous tall, because both genes are dominant genes. Here one gene is dominant, other one is recessive. So genotypically it is heterozygous tall and this one is homozygous tall. But from outer look, both are tall. So phenotypically both are tall. Filial, many times you wrote F1 generation, F2 generation. Filial means next generation. F1 means first filial generation, first generation. F2 means second next generation, second filial generation. Then we always draw our traits as our laws as the Pernit Square. We use Pernit Square. That is the simple checkerboard which is given by Pernit. Suppose I have taken here example of tall. So here this tall parent is crossed with the recessive parent. Gametes which will be formed, TT, here gametes will be formed. They're small t. So now you have to write these gametes. These are the male gametes, suppose, and these are the female gametes. So one side you will write male gametes and other side you will write female gametes. As you can see, here you will write male gametes and here we will write female gametes, and then cross them. TT, that will be TT. Now cross this T with T, that will be capital T, small t, then this will be, so all will be tall. But why this checkerboard? Why this is important? This representation actually give us the phenotypic and genotypic. Basically easy to study both phenotypic as well as genotypic characters with the help of this checkerboard. That is why we use this presentation. Selfing, जब आप क्रॉस कर रहे हो ऑर्गेनिज्म ऑफ द सेम जनरेशन, सपोज यू आर क्रॉसिंग पेरेंट्स, right? Parents you are crossing, like these are the parents. What will be that offspring will become, and that offspring will be this one. So you are crossing these organism of same generation. Now what is this? This is offspring and I am crossing, this is of same generation. So when you cross organism of same generation, that is called selfing. What is pure line now? Self crossing or self genotype for continuous 5 generations means once you get the same genotype for many generations. Like same genotype कैसे आएगा? Suppose now I have crossed these parents. What do you expect you will get every time? Every time either it's 1 or it's 2 or it's 3 or it's 4, 6, 7, any generation you will get only TT, till there will not be any small t will be involved. You will get the same generation. So that kind of crossings, that is called pure line. Once I will repeat it again because इसी में बहुत confusion होती है बच्चों को. Suppose I cross this, is there any kind of change you can observe? No, all the parents, they have same kind of genes, that is you will always get RR. So when there are same genotype is involved, स्पेसिफिकली इसको समझने का यही तरीका है कि सेल्फ जीनोटाइप, सेम जीनोटाइप इज इन्वॉल्व, दैट इज कंटिन्यूसली यू आर सेल्फिंग देम फॉर 5 जनरेशन और मोर देन 5 जनरेशन, then this will be same for that 5 generation. This kind of offspring, this kind of genotype is called pure line. इसीलिए इसको pure line बोलते हैं because there are no chances of वेरिएशन, जो कि हमारा सेल्फ पॉलिनेशन में बहुत ज़्यादा पॉसिबिलिटी होती है, राइट. नेक्स्ट इज द मोनोहाइब्रिड. मोनोहाइब्रिड इज when I'm going to talk about only one character, when in the cross, when you study of inheritance of one character, मुझे किसी प्लांट में सिर्फ और सिर्फ हाइट को स्टडी करना है, then that type of cross will be monohybrid cross. Now this is the height of the plant. It can be tall or it can be dwarf, but I have studied only one character. Which character? That is height, that is monohybrid. But अब मुझे 2 characters को study करना है, मुझे height भी चाहिए, color भी study करना है. That kind of crosses are called dihybrid cross. Height, for if it is dominant we will write tall, and if color is purple we write purple. These are the recessive ones, so we will write the small letters. So that is 2 characters here I am studying. One is tall, one is color. Trihybrid, now when I am studying 3 characters, मुझे height भी study करना है, color भी study करना है, axial position भी study करनी है, axial as well as the terminal position. So axial position क्योंकि dominant होती है, so we will take symbol of axial, that is why I have taken A. Tall is dominant, that is why I have taken T. Purple is dominant over white, so I have taken P. So here these are the recessive ones, so इनके सभी के small letters आएंगे. So 3 characters, that is trihybrid cross. So that is all about today's introductory lecture of principal basis of inheritance. We are going to study many important interesting things in this chapter, so stay with me with my lectures. Till then, take care, have a good day.